Hello students, welcome you all to a new video of Sunanda's tutorial. In this video we will discuss the whole chapter of cellular respiration. So we all require energy for our different activities in day-to-day life. That energy is stored in the organic food substances that we consume, such as carbohydrates, fats and proteins. Carbon bonds of these energy rich organic molecules, specifically carbohydrates, are broken down within our cells by stepwise oxidation process to release large amount of energy. So the energy is stored in our food, and finally they break down to produce large amount of energy in the form of adenosine triphosphate ATP, which act as energy currency. So we spend in currency for buying something, so our cell spend this ATP or adenosine triphosphate for doing different kind of activities in our day-to-day life. Okay, so such biological oxidations which takes place inside our cells, or carbon oxidation that takes place in all living cells, mainly carried by cellular respiration. The whole process is called cellular respiration. So what is the definition? It is a metabolic pathway. Actually it is metabolic as well as catabolic pathway. So altogether it is called as amphibolic pathway. Cellular respiration is amphibolic pathway, not only metabolic, it is amphibolic pathway by which cells oxidize organic molecules like glucose to produce ATP or adenosine triphosphate, which is the energy currency. Our cell use this energy currency to do different life activities. It is an integrated sequence. So the whole process is an integrated sequence of 4 major enzyme driven stages. What is enzyme driven? That means in all steps this process require in some enzymes. Okay, so what are those 4 major steps? One is glycolysis, which is the common one in aerobic as well as anaerobic. Then pyruvate oxidation, citric acid cycle, which is specifically takes place in aerobic one. Then oxidative phosphorylation. Okay, then actually before oxidative phosphorylation, electron transport chain, then oxidative phosphorylation to generate ATP. Okay, so what is the precise definition? It is defined as the process of oxidation of complex organic compounds to form simpler inorganic substances like CO2, H2O, with the release of some energy in the form of ATP. So in one line, it is the oxidation of organic substances by molecular oxygen to produce ATP or energy. Okay, so it is the oxidation of organic compound to form simple inorganic substances like carbon dioxide and water with the release of energy in the form of ATP. Okay, so it is fundamentally a catabolic process. It is biological mechanism where complex organic molecule like sugar, like glucose, are broken down into simpler one like carbon dioxide and water. I have told you that releasing chemical energy needed to produce ATP. Because respiration is key part of how body manages energy and resources, it sits at the center of body's 2 main metabolic pathway. So sometimes they act as catabolic pathway, sometimes they act as anabolic pathway. Okay, respiration mainly acts as catabolic pathway by oxidizing or breaking nutrients to release energy, and by breaking large molecules like carbohydrates, proteins, fats. But The main goal is breaking things down. The intermediate molecules created during respiration process are also used as building blocks of other metabolic pathways or building blocks to synthesize other essential compounds. So at the same time they break something and they are byproducts used as building blocks to synthesize some molecules. So we can say this is a catabolic pathway as well as anabolic pathway. Togetherly respiration, we can say respiration is a amphibolic pathway. The core function of respiration is breakdown of fuel to provide energy. So this is why this process is primarily defined as catabolic pathway. Okay. Now this respiration process can be aerobic as well as anaerobic. Aerobic, generally it takes place in the presence of oxygen, but in the absence of oxygen respiration also takes place, and that kind of respiration is called as anaerobic respiration. Then what is the meaning of respiratory substrate? Different kind of organic food substances like carbs, carbohydrates, fats, protein can act as respiratory substrate. But glucose or carbohydrate is the most suitable one, followed by fat and proteins. Okay, so we will discuss about respiratory substrates, respiratory quotient later on. At first, let us discuss about aerobic and anaerobic at first. So cellular respiration can be broadly classified into aerobic respiration and anaerobic respiration. Anaerobic respiration can be also termed as fermentation process. Remember, fermentation process. Fermentation process. Aerobic respiration, okay, takes place with the participation of molecular oxygen, and there is a complete oxidation or breakdown of glucose takes place to form carbon dioxide and water molecule. So at the end of anaerobic respiration you will get carbon dioxide and water molecule, and it involves the electron transport chain also, coupled with ATP synthase complex for energy production. Very large amount of energy, approximately 686 kilocalorie of energy, accounting a total amount of 38 molecules of ATP are released in this process, that means in aerobic respiration. It involves, this aerobic respiration involves cytosol as well as mitochondria for Krebs cycle and electron transport chain. In cytoplasm, glycolysis takes place, which is the first step of aerobic as well as anaerobic respiration. Then the product of glycolysis enter inside the mitochondria. Then the further processes takes place inside that mitochondria. Okay. Next, anaerobic one takes place without the participation of molecular oxygen, but it involves the incomplete breakdown of glucose. So here in anaerobic respiration, at first glucose first break down to form pyruvic acid or pyruvate, and then to ethyl alcohol or to lactic acid. And here there is no involvement of electron transport chain. It is less energetic also. Okay. Anaerobic respiration occurs in yeast, in some bacteria, even in tissues of higher organisms, in some tissues of higher organisms. Okay. So sometimes anaerobic respiration is termed as fermentation. But remember, fermentation always takes place outside the cell by extracellularly secreted enzyme or extracellular enzyme. Okay, so those anaerobic respiration which takes place outside the cell by extracellular enzyme is called as fermentation. Okay, it mostly takes place in yeast, in some bacteria, and in the in some cells of higher organisms. Okay, So it is the graphic representation of aerobic as well as anaerobic respiration. At first, glucose break down to form pyruvic acid, generating 2 adenosine triphosphate. When oxygen present, then it will follows the path of Krebs cycle, then oxidative phosphorylation, electron transport chain, then oxidative phosphorylation producing 36 or 38 ATP. If oxygen is absent, either they will follow lactic acid fermentation pathway or they follow ethanol or ethyl alcohol acetic acid fermentation pathway. Okay, so ethanol or ethyl alcohol fermentation pathway in case of anaerobic respiration. Okay, now let's discuss about the first step of cellular respiration, which is common to aerobic as well as anaerobic respiration, which is the fundamental 10-stepped metabolic pathway, which breaks down one molecule of glucose C6H12O6 into 2 molecules of pyruvate or pyruvic acid C3H4O3. This process entirely takes place in the cytoplasm of the cell, and it requires no oxygen and serves as the universal first stage of both aerobic as well as anaerobic respiration. This glycolysis process is otherwise known as EMP pathway or Embden-Meyerhof-Parnas pathway, who had major contribution towards elucidation of this path of glycolysis. Glycolysis itself is an anaerobic phase because it is independent of oxygen and not accompanied with the evolution of carbon dioxide. It takes place in cytosol or cytoplasm. It involves partial oxidation of glucose into 2 molecules of pyruvic acid, and it produces 2 ATP at the end of the reaction and 2 NADH. Okay. The product is the pyruvic acid, which acts as substrate for the next Krebs cycle or fermentation in case of anaerobic respiration. This whole 10-step enzymatic process takes place in 2 phases. The first phase is the preparatory phase or energy consuming phase, and the second phase is payoff or energy yielding phase. The first step of glycolysis takes place in the presence of hexokinase to produce glucose. From glucose 6-phosphate is formed. Second process is an isomerization reaction where fructose 6-phosphate is formed from glucose 6-phosphate in the presence of phosphoglucoisomerase. Then fructose 6-phosphate, from fructose 6-phosphate, fructose 1,6-bisphosphate is formed in the presence of enzyme phosphofructokinase and Mg2+ ion. Next one is the cleavage step to form glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate from fructose 1,6-bisphosphate. So it is a cleavage reaction. Then the next one is isomerization reaction, where dihydroxyacetone phosphate isomerizes to form phosphoglyceraldehyde 3-phosphate. The next step is an oxidation step, okay, where the oxidative phosphorylation of glyceraldehyde 3-phosphate takes place to form 1,3-bisphosphoglycerate, okay, with the formation of 2 NADH. Next step is from 1,3-bisphosphoglycerate 2-phosphoglycerate is formed, generating 2 ATP. Next, from 3-phosphoglycerate 2-phosphoglycerate molecule will be formed. Then from 2-phosphoglycerate phosphoenol pyruvate is formed, and then from phosphoenol pyruvate 2 molecules of pyruvate will be formed, producing ATP. So as this process takes place 2 times, from glyceraldehyde 3-phosphate to pyruvate and then dihydroxyacetone phosphate to pyruvate, there are 2 ATPs, all total 4 ATPs are formed. So there are the utilization of 2 ATP molecules, and the yield of ATP molecules at the end of the reaction is 4 ATPs and 2 NADH. But the net gain is 2 ATP because 2 ATPs are already required for this process. From this 4 ATP we can minus 2 ATP to get 2 ATPs only, which is the net gain of glycolysis reaction. Okay. So after glycolysis reaction, oxidative decarboxylation of pyruvic acid takes place within the in case of aerobic respiration, which begins with 2 molecules of pyruvic acid, which is the product of glycolysis from cytosol. Those 2 molecules of pyruvic acids travel from the cytosol into mitochondrial matrix. Within the mitochondrial matrix, the oxidative decarboxylation of pyruvic acid takes place to form acetyl coenzyme A, which is a 2-carbon compound. Then this acetyl coenzyme A involve or enter inside the Krebs cycle. Okay. So now the Krebs cycle. Krebs cycle is the second step of aerobic respiration, which was first discovered by Sir Hans Adolf Krebs in 1937. 1937 by Sir Hans Adolf Krebs, who was awarded Nobel Prize in Physiology and Medicine in 1953. Okay. This cycle is also called as citric acid cycle or tricarboxylic acid cycle, as citric acid or citric acid having 3 carboxyl group and all the earliest intermediates have 3 carboxylic groups. Okay, so that's why it is called as tricarboxylic acid cycle. Okay, as the first product, the first intermediate is having 3 carboxylic group, which is the citric acid, which is the first stable product of Krebs cycle. Okay. Now the biochemical pathway. So at first acetyl coenzyme A enters the pathway, and acetyl coenzyme A first binds with oxaloacetic acid to form citrate or citric acid. Oxaloacetic acid is a 4-carbon compound and acetyl coenzyme A is 2-carbon compound, so togetherly they will form citric acid, which is a 6-carbon compound. Then one isomerization reaction will take place to form isocitrate from citrate. Okay. Then the isocitrate forms the alpha ketoglutaric acid by the decarboxylation reaction. So here oxidative decarboxylation of isocitrate takes place to form alpha ketoglutaric acid. Here one molecule of carbon dioxide is removed, forming 5-carbon molecule, which is alpha ketoglutaric acid, and here one NADH is also formed. Next, from alpha ketoglutaric acid, succinyl coenzyme A is formed. Succinyl coenzyme A is formed by removing 1 carbon dioxide again. Okay, and here in this step 1 NADH is also removed. Then from succinyl coenzyme A, succinate or succinic acid is formed by removing 1 ATP. From succinate, fumarate is formed, and in this process 1 FADH2 molecule will be formed. Then from fumarate, malate will be formed, and from malate oxaloacetic acid or oxaloacetate will be formed, and then again it will receive one acetyl coenzyme A to form citric acid or citrate. So with this the whole Krebs cycle is completed. So at the end of this Krebs cycle we have 6 carbon dioxides, 4 ATPs, then 10 NADH and 2 FADH2. Okay, so it is the overall products of the whole Krebs cycle. So 4 ATPs from substrate level reaction. Okay, that means 2 from glycolysis and 2 from Krebs cycle. So all total we have 4 ATPs directly at the end of Krebs cycle. At the end of Krebs cycle we have 2 NADH from glycolysis, 2 from oxidative decarboxylation and 6 from tricarboxylic acid cycle or Krebs cycle. So all total 6 plus 2 plus 2, 6 plus 4, that means 10 NADH. So 10 NADH approximately provide 30 ATPs. Okay, so 4 ATPs directly, approximately 30 ATPs from NADH. From 2 FADH2 approximately we will get 4 ATPs. So all total 30 plus 4 plus 4, that means 38 ATPs. Okay, so at the end of Krebs cycle we have 38 ATPs. Okay, then the electron transport chain. So mitochondrial electron transport chain or terminal oxidation, final step, it is the final phase of aerobic respiration. During this phase reduced coenzymes which are formed like NADH, FADH2 released from different reaction stages of cellular respiration are oxidized by transferring their electrons to terminal acceptor, which is the oxygen or O2 molecule through chain of electron carriers. And okay, and finally O2 molecule is reduced to S2O. Okay, and finally the O2 molecule is reduced to H2O. This mechanism, this whole mechanism of oxidation of reduced coenzymes through electron transport chain is associated with phosphorylation of ADP to ATP. So the whole process of ATP synthesis coupled to respiratory electron transport chain is called oxidative phosphorylation. Okay, so where this whole process takes place? So it takes place in inner mitochondrial membrane and its cristae of mitochondria. Okay, this inner mitochondrial membrane bears numerous elementary particles which are called as F1 ATPase required for ATP synthesis in case of eukaryotes. In case of prokaryotes, this electron transport chain are located in plasma membrane as they are devoid of double membrane bound mitochondria. Okay, there are 4 complexes, actually 5 complexes arranged in inner mitochondrial membrane to carry out the electron transport chain. So the first complex is NADH COQ reductase, second one is succinate COQ reductase, third one is COQ cytochrome C reductase, and complex 4 cytochrome C oxidase, and the final one is the ATP synthase which is meant for the production of ATPase. And there are 2 mobile carriers, one is COQ and the second one is cytochrome C. So the electron flows from complex 1 to complex 4. Okay, and finally the ATP synthesis takes place at the end of the reaction. Okay, so the electron carriers are accomplished by transfer of electrons from a donor into an acceptor. Such oxidation reduction reactions which occur simultaneously are called redox reaction. Okay. So these reactions are catalyzed by dehydrogenases, reductase, oxidase. An electron carrier is thus alternately oxidized and reduced, forms a conjugate redox couple or redox pair. So at first the electron is accepted from the substrate to NAD+, then from FAD, then to FAD, then to COQ, then cytochrome B, then to cytochrome C1, then to cytochrome C, then finally to cytochrome A and A3. The last acceptor or final acceptor is O2 or oxygen molecule producing water. Okay. Then the last step, which is oxidative phosphorylation, which is coupled with electron transport chain, and the last complex, which is ATP synthase or complex 5, help to produce ATP from ATP. So this mechanism of electron transport chain dependent ATP synthesis is based on the most accepted chemiosmotic hypothesis, and it was first postulated by Peter D. Mitchell in 1961. Okay. So what are the key aspects? But at first, free energy which is released during electron transport chain. So we have to remember that free energy is released during electron transport chain. Electrochemical proton gradient occurs due to pumping of protons during electron flow. And the final one is the participation of ATP synthase complex, which is located within the same inner membrane of mitochondria along with 4 respiratory complexes. So during the transportation of electron, during the electron transport chain, it causes the loss of some free energy, which at some sites are used to pump proton from matrix to intermembrane space. So this proton pump occurs at 3 energy conservation site. Okay. So when the proton is pumped outside the matrix to the intermembrane space, finally it causes the proton gradient. That means high concentration outside in intermembrane space and less concentration in the mitochondrial matrix. It causes electrochemical proton gradient. So to balance or to maintain equilibrium, those proton again enter inside the matrix through ATP synthase system, and during their transportation ADPs are converted into ATPs. Okay, so it is the whole concept of oxidative phosphorylation. This ATP synthase complex, okay, ATP synthase complex, which is complex 5, have 2 components, F0 component and F1 component, F0 component and F1 component, which is projected towards the matrix side of inner membrane of mitochondria. Okay. So complex 5 or ATP synthase is otherwise known as F0 F1 ATP synthase, or F1 ATP synthase, or F1 ATPase. F0 component is an integral protein component which allows facilitated diffusion of protons down their concentration gradient back to the matrix to maintain the electrochemical balance, so that F0 component act as proton channel. The proton motive force is created due to the proton gradient. That gradient or that force drives the reverse flow of proton from intermembrane space to the matrix with the help of the channel protein of F0 component. And F1 component is peripheral protein component, which acts as catalytic component for the synthesis of ATP, which binds to ADP and inorganic phosphate, at its catalytic site inside the matrix, then the free energy of proton motive force released during the passage of protons through proton channel is utilized for the phosphorylation of ATP. ATP, finally ATP is generated from ADP and PI or inorganic phosphate. In the presence of the enzyme ATP synthetase or ATP synthase. Okay. So finally, with this, the 3 steps, glycolysis, Krebs cycle and electron transport chain associated with oxidative phosphorylation end. And after that we have carbon dioxide, H2O, water and ATP, energy in the form of ATP, which are the final products of cellular respiration, aerobic cellular respiration. Because in anaerobic cellular respiration, there is incomplete breakdown of glucose molecule takes place. Okay. After that, we have to remember certain things like what is respiratory quotient, RQ. You know about respiratory substrates. That means different types of organic molecules which can be broken down during respiration to make simpler substances. Respiratory quotient is the ratio of carbon dioxide produced to oxygen consumed. How much amount of oxygen consumed as comparison to how much amount of carbon dioxide is produced. So the respiratory quotient of carbohydrate is always 1, and for protein it is less than 1. For fats it is always less than 1, but for organic acid it is more than 1. In case of anaerobic respiration it is infinite. Okay, so it is about respiratory quotient. Okay, you have to remember that for carbohydrates the RQ is always 1, and for organic acids it is more than 1. For fat and proteins it is less than 1. For anaerobic respiration it is infinite. Okay, so with this I am concluding my lecture video here on the topic cellular respiration. I have tried to touch all the important points of respiration. So I hope this video will be proved beneficial for you all. And if you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's tutorial to get more important videos, more valuable videos for you for your examination. So with this I am concluding my lecture video here. Thank you.